Alan, Giuseppe, we're good to go.
Okay, great. All right, so welcome once again to the School of Materialist Research. And today we have, I have the great pleasure of introducing Giuseppe Longo and his talk, A World of Instructions, Computational Myths in Cognition and Life versus Alternative Paths for Human Invention and Understanding. Before I go ahead and introduce Giuseppe proper, I'm going to say a few words about the school. The School of Materialist Research is an informal graduate and postdoc level program that offers seminars and workshops that address the materialisms running through contemporary science, philosophy, art, mathematics, design, architecture, and politics. The school is an international platform founded by the Center for Philosophical Technologies at Arizona State University, Institute of Social Sciences and Humanities, Skopje, the Department of Architecture, Theory, and Philosophy of Technics at the Technical University of Vienna, which is in cooperation with the Critical Inquiry Lab at Design Academy Eindhoven. The school functions as a global online school for combining education, research, and mentorship to advance academic study at the intersection of the social sciences and humanities and the STEM sciences. And the spring and summer sessions are offered for free and open to the public, serving as promotional events that illustrate the programmatic commitment and style of work offered by the School of Materialist Research. Now I want to introduce Giuseppe, and Giuseppe is the Director of Research at CNRS, the Center for Interdisciplinary Center at Cavaillés. An adjunct professor in the School of Medicine at Tufts University in Boston. He's also a former professor of mathematical logic and later of computer science at the University of Pisa. He also spent 3 years in the United States at Berkeley, MIT, and Carnegie Mellon as a researcher and visiting professor, in addition to several months in Oxford, Utrecht, and elsewhere. He is a member of the Academy Europa since 1992. Giuseppe Longo also worked in mathematical logic and at various applications of mathematical logic to computer science. And in these domains, Longo co-authored more than 100 papers and a book on categories, types, and structures, published by MIT Press in 1991. And he is the founder and former director from 1990 to 2015 of Mathematical Structures in Computer Science, a leading scientific journal of Cambridge University Press. In this century, he's actually extended his research interests and work to the epistemology of mathematics and theoretical biology. And he's working, he co-authored a book on mathematics and natural sciences, the physical singularity of life, in 2006. And it proposes a novel approach to cross-foundational analyses in mathematics, physics, and biology. Its consequences for more specific theorizing in biology are in a joint book with Maël Monteville titled Perspectives on Organisms, Biological Time, Symmetries, and Singularities. And this was published in 2014. And he's also edited a special issue in 2016 titled From the Century of the Genome to the Century of the Organism, New Theoretical Approaches, which focuses on new approaches to biology of organisms, integrating the evolutionary principles in the understanding of ontogenesis. And he actually is also the current chair of the Association of Friends of the Thunberg Ars Industrialis Generation. I'm not sure if I'm pronouncing this correctly, Giuseppe, but he's also incredibly busy and active in a number of European scientific networks that are addressing environmental issues on a variety of fronts and scales. So with that, I want to hand it over to Giuseppe Longo. So thank you for joining us, Giuseppe.
Well, thank you very much. Can you hear me? Is that okay? Is the sound okay? Fine.
Yes, it is.
I'm pleased to be back in Arizona. It was 4 years ago, I think. but then I was physically there. Let's now try to communicate with these new tools, which are quite fantastic as a supplement of physical and presential activities that I hope will be resumed very soon. Well, I will try to hint to a perspective that goes back to the construction of sense, in a sense, in human activities, and criticize some distortion of this from what is the center of our being in the world, but also the new uses we can practice of machines and relations to other humans by these tools. Perhaps it's an abuse, but an early configuration of sense was our invention as humans when we started interpolating stars. That's a very mathematical gesture, but then it was a locus not only for practical reference in positions, but most likely for the reference to divinity, to beyond the world activities, the creation of myth. Now, this went on the walls, this way of relating to the world by imagination, as probably in a very high moment when humans started drawing abstract structures and giving meaning to them. In Lascaux, there are several paintings of bisons and horses. In my view, the most interesting ones are those obtained by just drawing borders. You see, objects do not have borders. There are singularities, differences in colors and frequencies of light, but not borders. And we humans, we invented borders to delimitate and communicate to other humans. Indeed, only a human can understand that this is a bison. You need the common gestures, and in language, you do express what it actually means, a drawing. This idea of having borders went into mathematics in the most fantastic invention of Greek geometry, the invention of borders. Because you see, what's absolutely crucial in Euclid's approach is what was later explicitated as definition beta, but it's present everywhere. Namely, the fact that the line is a length with no thickness. I mean, no thickness doesn't exist. Anytime you draw, there is a thickness because you need to see it. Yet, in abstracto, inventing the border is the only way to do a sound geometry of surfaces. Because if you have a surface with a border, which is the actual measure of the surface, so the notion of border is the real structural foundation of Greek geometry. It's very, very pleasant to see that for 1 century, Euclid's only been referred to as the inventor of the axiomatic approach, which is true, is half of his job, of the school of Euclid, half of the job. But the other fantastic idea that only Greek geometry has, not the Chinese Louis 7 centuries later, only Greek geometry sets this statement that the border has no thickness. That's the invention of a crucial notion for doing geometry in the purest sense. And indeed, the lines are all what exists in Greek geometry. By translations and rotations and symmetries, theorems are proved. There are trajectories. Indeed, in the axioms, you could say traces produce. That's what it means to be a border. It is a trace, it is an intended trajectory. Then later, this will be specified by continuum in various ways. Descartes, Newton, and Cantor will elaborate for that. But the early invention, I see it in continuity with this invention of border as early as Lascaux, some 20,000 years ago. Now, there is a major distinction to be made that we owe to René Thom between, in figures, what matters for drawing, what matters in the appearances, what René Thom, a major mathematician of the 20th century in France, called sciences, perhaps in English, to be compared with prégnance, which means what is pregnant, which is meaningful, which is rich of meaning. So, for example, this beautiful little statue, a present by Xingwei to me, not the original. He gave me a present of a copy, unfortunately. It is an Etruscan sculpture. A sculpture is in bronze, where the artist gives us only what matters. In this horse, in this man, there is nothing more what it matters to express, what is the artistic sense of this horseman chariot. But we do that all the time. Look at the caricatures by Plantou of Chirac. No machine would recognize Chirac in this caricature because the artist has been giving us only pregnancies. What is pregnant, what is meaningful, is gesture with the nose, the structure of the chin, the moving chin. It is movement that it expressed, in a sense, like in the artist, in the Etruscan artist, that gave us only what is meaningful in what he wanted to represent. Indeed, we animals, not only humans, we do recognize only caricatures. Namely, when we see a face, the first thing we see is the expression. It is exactly what a good painter can communicate, the expression. What matters to us are not the forms recognized also by machine, but the expression, which are movement. If you meet someone after 40 years, a friend at school, first girlfriend or boyfriend, you don't recognize the details that entirely change it, but you recognize the way of the folding under the eyes, the smile, the movements that were meaningful to you, that keep being meaningful. The animals recognize first the postures. They recognize the aggressive attitude of fear before recognizing the form, the saliences, les saliences. Indeed, I insist to say that we only recognize pregnancies. Then later we can describe also the saliences. That's radically different from machine recognition. Typically, these two girls, these two ladies, were presented to all machine learning techniques for recognizing visual recognition of faces, and they were not recognized. Why? Not only we understand there are two women, but we understand they are quite sad. Perhaps they were bored by all that exercise with all possible AI system for facial recognition. I insist, in order to make the difference between what it means to recognize the silences, namely the forms, and the pregnancies, namely what is meaningful to us. And again, in these two girls, we first understand their mood before even understanding that they are people. Now, we are embedded in a myth of the identification with nature, with machine, which is an old one. It goes back to the scientific revolution. You see, the scientific revolution was led by fantastic mathematics and physics centered on celestial mechanics. This was not in continuation with what was happening at the time in Italy, in particular, in Renaissance, where a deep naturalist, by analyzing and studying God in nature, did not identify machines to nature at all. Leonardo, typically, as I will show and hint, I've been writing about that with my daughter, who is an historian of art, in particular working on Italian painters of the Renaissance. And I will try to hint the difference and how we got into a radical mechanicism that is still now heavily affecting scientific knowledge, in particular in AI and biology, molecular biology. Leonardo loved complex disorder structure. Leonardo made only 15, 20 paintings, but hundreds of drawings, where the interest was not only artistic, he was a fantastic painter and drawer, of course, but he studied nature. He was an immense naturalist. Here he is trying to understand movement and complex structures, disordered structure that get to some order. Even more so, in these two turbulences, the air turbulences and water turbulences. The details are drawn in order to understand. He calls them as ephemeral phenomena. He likes the contingency of these phenomena. And he has a fantastic remarks. He says this kind of movements of air and water must be of the same type, because in order to draw them, I have to do the same gestures. He was so good as a drawer that he understood nature by looking and working at his own drawing. And he understands something very deep, which is totally correct, even mathematically. The turbulences are of the same kind, and he understands it by this remark on how he was drawing. So he was interested in this kind of phenomena. He makes really remarks of a major scientific thinker, like also on the foundation of mathematics. He goes against Alberti and claims that, like in Euclid, a line is a moving point, a plane is a moving line. In the sense, the opposite of what is said often today after Cantor, but was also Alberti's position, that the line is made out of point. No, the line is a moving point, which is very different. The plane is not made out of lines, is a moving line. And so he theorizes, that's in Italian, the mind of a painter must become the mind of nature itself and make itself interpreter between nature and art. So this is very different from what will come out to be the paradigm for the scientific revolution, where they see nature as made out exactly in the triangles, circles, and other geometric figures, regular geometric figures of Greek geometry, as Galileo thinks, even more, I will insist, Descartes and Francis Bacon. That's where really mechanicism begins. Again, Leonardo and Cardano, the other one I will mention, were not mechanicists at all. They were interested in those kind of structures, turbulent structures that are proper to ephemeral phenomena, contingency of diverse dynamics, nonlinear dynamics, we would say today. And at the same time, Leonardo was a major inventor of machine, but it was a completely different activity. Indeed, he invented the machine drawing by showing how they compose by small pieces, one attached to the other. And he was first drawing the pieces and then drawing the machine made put all together. And he had some sort of a bicycle, an helicopter, and lots of other things. Guns, drawing lots of guns and realizing them for the defense of Milan. So the mechanicity, which was proper, let me show one more thing about Leonardo, the order from chaos. That's a beautiful Sant'Anna, the Madonna, and the child. You see the movement there, this kind of spiral going up. It's both, there are two paintings, one in Louvre and the other in London. It is obtained from chaos, namely in the back of the preparation of the painting, in the paper preparing for the painting. He first draw lots of line, mixing up here in a chaotic way. Then from there, he started. That's recto verso. That's one side. That's the other. On the opposite side, he started drawing the lines emerging from the movements he had in the back. And of course, he was using the same piece of paper for something very different. Probably the paper was very expensive, so he was safely using it. So that's really kind of order from chaos, which became so much in fashion in the last 50 years. And look at here, the stressing of the dynamics. That's a man in a battle. These are studies for the Battle of Anghiari. And that's a man turning suddenly to the left, and he said continuous in order to express his state of mind with the lion in continuity with his head. And look at the horses. What interests Leonardo is movement, is turbulence, is the activity. And then in some cases, you have some order emerging from this. Cardano, 50 years later, we are in 1550, also thanks to material structures of nature, without any dualistic attitude. He makes, for example, he was a doctor and a mathematician, invented complex number, the Cardan joint, you know. He observes that I know that souls are immortal, but I do not know how this works. That is concerned because, again, for these people in the 15th and 16th century in Italy, there was no split, matter and soul. There was a unity, type nature was in. And so where those turbulences were part of the observation to be made, and Cardano is interested in the formation of clouds and the chaotic dynamic of clouds, the diffusion of winds. So for him, these unpredictable phenomena were an object of analysis. By the way, we started with we have 4 mathematicians who started a small group called Girolamo Cardano. We have a web page where we take lots of initiatives, revitalizing this attitude of knowledge of nature, which is radically materialist, because there is no dualistic split there, and which is highly motivated by sense, by the construction of sense. All the figures of Leonardo are the results of the imagination of a configuration of sense, emerging sometimes from chaotic situations. But then the scientific revolution came. Clocks and watches are the paradigm. Of course, they do fantastic work. They invent modern physics and modern mathematics, no doubt. But the attitude is the search for mechanicity in all aspects of nature. Galileo is a physicist, so he doesn't talk very much about life. But again, for him, the construction of machine is a way for understanding also the mechanics of the universe. Of course, what happens is the fantastic mix of observation, theory, and practice, which is the methodological revolution of the scientific revolution. Descartes and Bacon are much more radical because they discuss biology and life, and for Descartes there is no doubt. Animals are machines. Also, our body is just a machine. There are just movements of the composition of parts. They are clockwise machines. There were fantastic clocks invented at the time, of course, and there were lots of pumps and other gears and pulleys that did a lot of things, and the identification was radical. This was a change, a major change with respect to the attitude of great like Leonardo, Cardano, and the others of the Renaissance, Italian Renaissance. Not to mention Francis Bacon. Francis Bacon is almost a parody, to be honest. It is the reference for all, the guys in biotechnologies, because he theorizes that we must drive nature as a machine through action. So we must acquire full control of the ecosystems, animal, plants, and drive them to our will. That's the attitude, for example, of a recent book, 2017, of which I'm writing a review by Jennifer Doudna, the Nobel Award of 2020 in biology. Her plan is to program evolution. There's a software, amount of software, and like Francis Bacon claims, we will drive the ecosystem with our tools, CRISPR, and so on. These tools are fantastic, like the tools that the guys of the scientific revolution gave us. The vision of nature, in particular, vision of life, is miserable, to say the least. It's absolutely miserable. That's what I'm writing in my review. It should be read in a week. If you are interested, just let me know. So the turning point is really the end of 16th century and the 17th century. So Galileo, Descartes, and Bacon are not interested in the ephemeral, the turbulences, the contingency, which were instead at the core of the interest, as I mentioned before, of the previous naturalist. They look for stable invariant phenomena. which of course is very important, and they made fantastic things with this view. A major radical dualism is introduced. So on one side there is the soul, and the other there is matter. Even Galileo, again, is really a physicist. It doesn't study life, except that he's very sensitive to the diversity of life. Yet, the only technical work he does in biology is the invention of allometry, which is a beautiful invention. Allometry is the analysis of the change of scale and the changes of the structure of the bones, typically. So he observes that if you increase the length of the bones, then the section of the bones should not increase like the square, as it would be for maintaining proportions, but it should increase like the cube, because the weight increases like the cube. And it is so, it is quite fantastic. So if you take the bones of a mouse and the bones of an elephant, the observation is perfectly consistent. The bones of an elephant have a section which is much bigger than you would expect in proportion, simply because it has to go with the cube of the length to support a weight increasing with the cube. That's a very nice remark, still today valid, but is a mechanical property of life. The other properties are not of his interest. Not to mention Francis Bacon, as I said, is really the paradigm of the worst technoscience we have today. This mechanicist attitude did continue, and the best representative are probably the major mathematicians, Lagrange, Laplace, Fourier, because they mathematize, they make clear what it means to be mechanistic. What it means? It means that you can obtain anything by composing elementary and simple components. You know, any machine, even our very complicated computers, you construct them by taking elementary and simple components, and then you make something very complicated, even a computer network. By adding more and more elementary and simple components, you get to the complication of the computer networks. The mathematical counterpart of this is the idea they had that any reasonable system of equations, even nonlinear equations, may be approximated linearly. And they did fantastic stuff with that. I mean, Fourier series are very important. But the main conjecture they had, I didn't quote Lagrange, but he belongs to the group of major mathematicians. The main conjecture they had was they could decompose any deterministic dynamics, however complex it is, nonlinear and so on. For example, those representing turbulences, even though they were not thinking to turbulences, but for example, they were thinking to the planetary system. As soon as you have one sun and two planets, you have a nonlinear system, which is difficult to solve, but they thought it could be approximated linearly, so decompose it mechanically. Well, this is false. That's really the major defeat of mechanicism that has not been enough integrated in our culture. In 1892, Poincaré showed that as soon as you have one sun and two planets, because of interactions between the two planets, the nonlinear system does not have an integral solution, nor most of the time, almost everywhere in mathematical terms, it can approximate it by Fourier series, Linstedt Fourier series in particular. That's an extremely important result because it shows that, okay, you start taking a sun, then one planet, then from Newton equations, you derive all Keplerian properties of the ellipses, which is fantastic. It's the great achievement of the very simple system of equations by Newton, where from mathematics, you derive Keplerian properties. Great. Newton knew that if you add an extra planet, because gravitation is universal, the interaction of the two planets destabilizes the system. And he understood that there was a major problem in proving that the system is stable, in particular in proving that there exists a solution to the system of 9 equations on a plane presenting the 3-body problem. And he gave the only solution we have today to have stability forever, namely said, God, time to time, gets back and puts the planets in their right orbits. I insist, Newton was perfectly right. He understood without having a theorem that there was a major problem. So Poincaré showed, indeed, the system is chaotic. Of course, in the long term, it's not the same chaoticity as the turbulence of water, or the throwing of dice, but there is no integral solution. There is no provable stability. Indeed, the system is unpredictable. Well, in the case of the solar system, it's a matter of 1 million years. So we shouldn't worry too much. Just the Earth is 100 million years, the unpredictability. But yet, it is a chaotic system. Then about the same times, physics departs even more radically, because in quantum physics, indetermination and entanglement are really non-properly mechanical properties. I'm not getting into the details. But even relativity theory, thinking of continuous deformations of space-time doesn't belong anymore to a mechanistic tradition. I mean, the imagination of the scientists goes very far from Laplace and Fourier, not to mention Descartes. Unfortunately, mechanisms went back entirely after World War II with molecular biology. And the founding fathers, who were excellent theoreticians, very explicit, say that explicitly. The cell is a Cartesian mechanism, you see, autonomous, exact, independent from external influences. Necessarily, stereospecific molecular interactions explain the structure of the code, a Boolean algebra like in computers. Amazing, you see. The macromolecular interactions are interactions between huge molecules with immense enthalpic oscillations that in chemical physics were handled statistically since the 50s. Because of the Brownian motion, the chemical instability, the changing coefficients of affinity according to temperature, context, pH, make the interaction stochastic. They must be given in probabilities, and these probabilities depend on the context. Yet, in order to force the vision of a cell as a Cartesian mechanism, these guys invented that the macromolecular interactions are exactly like gears in a clock, the key lock paradigm, which was transferred from small molecules to big molecules, just for ideological reasons, in order to force the mechanicity of cell and, of course, the organism. And then, of course, we go back to Bacon. If we could create better human beings through the addition of genes from plants or animals, why deprive ourselves of them? What is the problem? It's a matter of a combinatorics of gears. I change a gear, I change the letters of the alphabet. Yes, it's a writing. The surprise is that the genetic specificity is written not with ideograms as in Chinese, but with an alphabet. François Jacob. I told this joke in China. Unfortunately, it was not a joke. And we reached the compromise. The Chinese have their DNA written in ideograms. This was our compromise. It's amazing. The alphabet allows editing, it's exact writing. Then you change the letters and you do what? You do what? The problem is the network of interaction, of flexible, modifying, context-dependent interactions allow only time to time to see that when you change something will happen. And it may have some regularities, but in no way you drive an organism by acting on DNA. Since 50 years, they've been promising us to cure the monogenetic disease. The monogenetic disease are 15, 20, no more, because they're very rare. Monogenetic means that we really know that this is due only to the mutation in one place in the DNA. That's what monogenetic. There are very rare diseases. There's only one that we cure. Even not the monogenetic diseases have been cured with the idea that life is a combinatory of alphabetic letters. Okay. But this mechanistic view of life continues. Gilbert, a major proponent of the Human Genome Project, several billions to decode. It's very important to know the chemical structure of the DNA. Very important. I mean, the point is to see what it really does in an organism. Once they coded the DNA, we were able to transfer it on a CD-ROM and say, here is human being, this is me. You have caught the first glimpse of our instruction book, previously known only by God. And so personalized medicine was proposed as, you know, you go to a hospital, they decode your DNA, you put it on a DVD, they send it in various departments. In time, perhaps you, instead of having doctors examining your decoded DNA, you may have machines, good AI machines like Watson, and then you will have a medicine with no patients and no doctors. And this is called personalized medicine. Again, the DNA is very important. It's the physical-chemical trace of evolution. The point is to understand it as an internal constraint to the molecular activity, to largely chaotic molecular activity. You know, almost every gene is expressed in almost every cell. It depends on probabilities. Its amount of expression, that is a statistical matter. It depends on the context, on the pressure, on torsion, on the chromatin, namely on the structure of DNA. DNA is a constant, is not the driver of an organism. As I said before, the key point is this mechanistic view that is still leading most molecular biology. Fortunately, there is increasing dissent. The core principle, as I said before, is the myth, which is immediately and provably wrong. I mean, anybody knows that at normal temperature, huge macromolecules have huge oscillation, change their structure, interact stochastically. And again, this must be given in probabilities depending on the context. But this engineering metaphor is dominating. The central dogma, I will not get into detail, and the central dogma is there, namely information DNA fully guide, or perhaps, yes, I can say something about that. It is still taken up in major texts. The key lock and hand glove paradigms, molecular biology, they allow the transmission and elaboration of biological information. They are the gears of life. And a small minority keep insisting it's not like that. Proteins never do fold in a particular shape, but rather remain unstructured and disordered. In mammals, about 75% of signaling proteins and half of all proteins are thought to contain long disordered regions, while about 25% of all proteins are predicted to be fully disordered. Many of these intrinsically unstructured proteins are involved in the regulatory process and are often at the center of large protein interaction networks. It is the cell, the partition in the cell proper to eukaryotic cells, the global structure that allows the stability of extremely complex network of molecular interactions, which include, of course, this huge, extremely important molecular complex, which is the DNA, which are the chromosomes, that are used in order to renew the protein structure and the chromosomes themselves. Yes, I should go into this because you see it in any high school book and also at university books, in particular in medicine, the so-called central dogma is stated in the 60s. You see the gene entirely gives the structure of RNA, then the proteins, which goes to the phenotypes. to shape your internal ears. That's been slightly complicated in more recent textbook, but that's what you see. Still, the leading role of a string of letters that should contain the code of life. The instructions once known only to God, as Collins observed. Collins was the head of the Human Genome Project and is now the head of NIH, National Institute of Health. Then one may wonder, what is a gene? The notion changed 5 times in the 20th century. Looked at Collins in a paper of 1999, claims that he was expecting in a project he was leading, the coding of DNA, was expecting 80,000 genes, because he still believed the association of one gene, one enzyme, proposed in 41 by Biddle and Tatum. Then 2 years later, they will acknowledge, well, no, there are 20,000. But by the way, in the 99 paper, Collins is very proud of the fact that we have 80,000 genes, because C. elegans, which is a small worm of 1,000 cells, microscopic, you cannot see it, it's very, very small, very simple. It was already decoded that 16,000 genes. So Collins, there is some pride in saying, well, look, C. elegans has 16,000 and we have 80,000, of course. Then 2 years later, well, no, we have 25,000. Actually, 3 years later, they went down to 20,000 genes. The genes of C. elegans are stable, 16,000. So the difference between us and C. elegans is 4,000 genes. But what is a gene? In 77, overlapping genes were discovered and hidden under the carpet. Now, fortunately, more and more people are understanding their major role, for example, in SARS, COVID-19, because there are a lot of overlapping genes which gives major flexibility according to the context, to the activity of the virus. But that's a side remark. Overlapping genes, what? It means that if this is considered to be a stop and the beginning of a gene, there is gene production which overlaps, uses part of the segment of DNA, it overlaps a border, a stop, for example, it uses another part, and the composition of molecules resulting from all the process goes from DNA to RNA to the proteins, goes by using pieces and fragments that cut any border of what are presumed to be a gene. Namely, there is no stable notion of gene. The DNA is used in a variety of ways according to the context. Yes, let's go to the modern contemporary issue. As you all know, probably SARS-CoV-2 was immediately decoded on January 17. Which again is very important. I mean, in a virus in particular is little else. So it's very important to know its RNA structure in this case. But in per se, the knowledge of this incredible molecular complex, the DNA and the RNA in a cell, is very important, provided that is inserted in a good way in its context, is not considered the leading coding machinery for instructing life. But then what does it do in a context? No idea from the genetic structure, because it depends on the organism, even in the ecosystem, because symptoms can range from mild to mild to severe, to having to go to hospital, to being on life support, to death. Some people recover quickly, others take many months of rehabilitation to recover. Some never seem to fully recover. Lungs may be so scared they will never fully recover. Neural damages are, in some cases, stroke, brain hemorrhage, and memory loss. As I mentioned before, it seems to have a high number of overlapping genes, and the way the overlapping allows to interact with the proteome depends on the context. Now let's get into the debate, today's debate. Is this a zoonosis or is that being artificially created in a laboratory? Well, in 2015, there was a very interesting book by Jacques Morin, a French colleague, and Figuier, where they showed, in reference to many papers from '93, the explosion of epidemics. The explosion of epidemics. The black ones are those that come back every year, the recurrent ones. The others are epidemics that normally stay local. Then, of course, there may be reasons why suddenly an epidemic may become a pandemic. The point is that 70% of these are zoonoses. What it means, they get to humans from wild animals, most of the time passing through breeders, I mean, animals in agriculture. And this happens mostly in those countries where deforestation is followed by installing agriculture and breeders at the border of areas that had undergone massive deforestation. This means Latin America, some areas of Africa, part of India, Indonesia, 70% of cases. So this book in 2015 was really trying to set an alarm of this explosion and closing and analyzing these vast majorities of zoonotic origins of epidemics of all sorts. It's very often mild, or some other cases like Ebola, very strong, so terrible as it kills in a few days after lots of sufferings. It doesn't spread, it doesn't go very far, it stays local, of course. You have no time to travel and infect the others. But in other cases, it may spread, and we go from an epidemics to a pandemics. Or was it artificially edited? You see, we are in a situation, we have been giving fantastic technologies in the hands of guys who pretend to guide evolution, to drive evolution by acting on DNA and RNA. These technologies came from outside biology. They came from chemistry, crystallography, electronics, even informatics plays a major role. They are in the hands of genocentric fanatics who think that can play as they wish on DNA and they will guide perfectly well an organism like GMOs in any context. In a paper, we developed a closer comparison between the genocentric perspective and the geocentric perspective, the Ptolemaic perspective. I don't get into details, but perhaps in these circumstances, one can make this further analogy. We are in a situation, something like giving a rocket capable to reach Mars to, Ptolemaic people in the fantastic Arabic culture of astronomy. They were excellent observers, but they had the Ptolemaic views. Suppose they could launch rockets with their theory, without considering, of course, the speed of rotation of the Earth and many other parameters we have today. They would send their rocket on the next city, somewhere else, surely not to Mars. We are in this situation. People with the genocentric view have in their hands technologies totally inadequate to their scientific ignorance. So when I'm asked in our group of concerned scientists about the principle of precaution, my first principle of precaution is a scientific one. When I understand that there is a discipline where the understanding is not scientific, because there is no doubt the language of information, programming, and so on, adapted to DNA in ignorance of what information programming is, without any sound, robust scientific definition of information, programming, or gene, is sufficiently bad to say, well, by scientific precaution, I do not take the products of these guys. If they propose GMOs, I don't need to make experiments to see that they give troubles. A priori, I can say their scientific frame is totally wrong. So I don't buy those products. Central dogma, key lock metaphor, they are provably wrong. Again, no way, the DNA is very important. It is continually used by the organism, but how is it used? This is a beautiful example of the cave fishes. They are blind fishes living in caves. There are about 100 species. As they are sufficiently close, hybrids are possible. The hybrids have eyes. The eyes is still there. Probably it has been mutilated, or well, we don't know exactly what happened, but it is not used. So those fishes do not have eyes, and that's very useful because there is a vicariance by the brain. The parts of the brain which were used for vision are used for increasing the sensitivity of the skin to vibration. So the fishes are very sensitive to water vibrating back against walls, and they manage in the complete darkness to move around. So the DNA is being used differently according to the context. This is not a denial of the relevance of having all the knowledge today we have about DNA and its molecular structure. You know, also in Ptolemaic astronomy, the Arabic astronomers, which were immense, I mean, they invented trigonometric geometry, lots of algebra, was also motivated for discussing planetary movements. Well, they was mostly motivated by, you know, an abstraction of arithmetic from India and the abstract Greek tradition. They invented the notion of function, of variables, but they had the totally wrong theory for the movement of the planets. Yet they constructed such detailed such detailed books of the tables about the positions of the stars and the planets that they wrote those tables that are known as Alphonsine tables that were constructed in the Catholic Spain at the end of the 15th century, which were used by navigators and successfully used. And Columbus could get to Americas using those tables, which were invented in a totally wrong frame, but with data which were correct. So we are getting lots of correct data. There may be some useful applications. Nonetheless, the theoretical frame was totally wrong. By the way, the geocentric Ptolemaic frame is not wrong on grounds of observations, because any finite number of position on an ellipsis around the sun may be approximated by enough epicycles around the Earth. It's a matter of decomposition of series. So it's not the observations that deny the validity of the geocentric system. It is the principle of inertia, the invention of the first conservation principle by Galileo. The principle of inertia, again, the first and most fundamental principle of conservation in physics, which is a symmetry in the equations. So it's absolutely crucial, like energy conservation. That is what first was inertia. Forbids epicycles because planets should go back and forth suddenly against inertia. So they change their perspective, allowed to understand the use of the principle of inertia, which gets into Newton equations and then provides a unified explanation of falling apples, moving planets, and that's then the beauty of physics, of constructing a sound, robust theory. Personally, I work with several colleagues to new principles. Of course, the first one is that macromolecular interactions are stochastic, they must be given in probabilities, and these probabilities depend on the context. That almost all most common phenotypes use almost all DNA. the cell and the organism and the ecosystem strongly canalize, constrain, and select macromolecular interactions, starting with gene expression. So we have to start from the organism and its relation to the ecosystem for the understanding also of the fundamental role of the DNA. No way to think that you will guide the plant, the animal, by acting on the DNA per se. So we are moving from the left to the right, where the level of interactions are cross structures from the molecular level to the organismal, to the phenotypes, and so on. I would like to suggest some readings. The beautiful book, which is an history, but also philosophy of biology of the 20th century by my dear friend Evelyn Fox-Keller. Evelyn is somewhat physicalist. I mean, she believes that the ultimate explanation at the molecular level, but she's strongly against the genocentric perspective. Then the other books, I think Couppier Canzonigo is translated by Niall. And Noble is a very interesting one. And then this paper by two former students of mine, now independent researchers and very strong researchers, I think is really a major novelty in the understanding of organisms. And here are some more writing and books that either you can download from my web page or see the references, exact reference for the book. My intent in this presentation was to see how an understanding of nature, which was married with beautiful artistic expression, which was unifying God and nature, mind and matter, understanding them at once, and also paying major attention to complex phenomena, ephemeral turbulences, was the richness of the early scientific thinking of the scientific revolution that was in the middle of the Italian Renaissance. Later, there was a turning point. I'm not an historian. I don't know what happened exactly, but perhaps the success of machines, I don't know, something happened in the end of the 16th and early 17th century, why suddenly the wonderful clocks, clockwork machines and pumps and other things they've been constructing were directly identified with nature. And this continued, partly in physics, but physics took also other directions, very rich directions, hydrodynamics, thermodynamics. There are original ways of thinking, quantum physics and relativity theory. And suddenly there's been a resurrection in biology via mainstream molecular biology. Fortunately, there are many who follow different directions, but mainstream molecular biology, which I think is a major problem we have to face, and in order to rethink nature in terms of its radical materiality and contingency and richness of dynamics that I try to favor. Okay, are you there?
Yes, we are.
Yep, we're here. So I'm imagining that's, should we move into questions then?
Yes.
Okay, awesome. So we have about, thank you. Well, first of all, thank you, Giuseppe. That was an amazing talk. If you could maybe stop sharing your screen. Is that possible?
Yes.
And then
How can I? Oh, yes, I see. Here it is.
Great. So we have about half an hour for questions. And as we usually do, you can either raise your hand or put your question in the chat and we'll get to you. So does anybody want to start off with a question?
I'm open also to make connections, say, with AI, because I think the formalist mechanical views has had two major consequences. The classical views in AI that are now changing because deep learning, somewhat different frame, and then molecular biology. These are the two aspects of the mechanistic views that still have a major role in technologies.
Yeah. Wait for folks to formulate their questions. Devor, do you have something?
Yeah. Okay, hello, can you hear me? Does it work?
Yes.
Yeah, okay. Okay,
Yes.
thank you, Mr. Longo, for the beautiful summary. I've been dealing with similar questions in my PhD on the history of types of knowledge during human evolution. And I wanted to add, how should I say, I want to add an idea that maybe the genocentric view was not backwards. You know, it was not a step back to the mechanistic perspective, but it was simply the first time that we saw the genes. And the initial step to see genes is a mechanistic model. It cannot be different, you see. So the resolution of reality was only possible in the 21st century to go down to the gene level, so to speak, you know, and then it was modeled, and it was not a backward step, but it was simply too late to get to this. So it came later than the other developments in science. Did I make it?
Yes, but the chromosomes were isolated at the beginning of the 20th century.
Yes.
That's where it begins the genocentric. But it really gets to be mechanical after the decoding, also because new machines were dealing with information. So the information programming paradigm was imported by the fact that at the same time, in the 50s and 60s, machines were not only, well, they had been invented slightly before, but they were very successful. They were starting to pervade science. Yes, it was later when we could get to this level.
Exactly. So, and what
but in order to analyze this level,
yeah, exactly. What I'm saying is, so to speak, you need to get to this resolution, right? And the first step of even seeing something would be, so to speak, to get to this resolution, then to put it into a sort of mechanistic model, and then based on this model, you can go to the next step, to processual thinking, as you are suggesting. So you see my point. So it was not a step backwards, but it was just biology only reached this moment through technology. First.
Yes, yeah. I agree. I mean, there is a fact that we suddenly could go so far, and that's very important. I insist. Knowing the chemical structure of the DNA is very important. Look in evolutionary theory, in theory of evolution. As they have a beautiful theory, which is 200 years old, it is a theory with two major branches that disagree, but still there is an open debate of principles, robust principles that are confronting each other. They made a fantastic use of the knowledge of the DNA. This knowledge helped to reconstruct phylogenetic paths as it could never be done before, because they have a beautiful theory. In absence of a theory of organisms, suddenly this level was the only one analyzed, and it was considered to be complete. We know everything by acting at the molecular level. This was the mistake, I mean, the conception with major consequences. As some of my colleagues say, is that the genocentric perspective is too big to fail. They've been given too many jobs, too many grants, too many projects, too much money to say, oh no, we should change perspective. They cannot. They cannot.
Okay, thanks.
Anybody want to ask a follow-up?
Maybe I could jump in here.
Yeah, Alec.
Thank you, Professor Longo. That was very interesting. I would just jump in maybe here after Davor's question, and kind of a connected question. But about the way you were talking about DNA as a kind of mechanistic metaphor. What do you think about the RNA world hypothesis? This idea that, before DNA evolved, there was RNA, which is a kind of, because you showed us the central dogma. It goes from the code, to the RNA, which is in between, that then produces the proteins. But the RNA world hypothesis is the idea that the RNA world preceded DNA before genetic or DNA-based organisms. There was some sort of RNA world, which was I find that this idea very appealing, because then it seems like this idea that the code kind of emerged as one of these so contingent biological solutions for creating more variety. The RNA was on its own was limited because it couldn't, kind of the same way that recombination appeared on the scene further down the line. Recombination and sex life appears on the scene in order to create more randomness, more contingency. And it seems to me that the DNA if you go back even further, and if this RNA world hypothesis is right, that the DNA is also part of this process of differentiation in order to produce more variety. I don't know what you would make of that.
Well, I'm very careful not to get into the debate on the origin of life, because I think we know too little. It's too speculative. What I can say is that the role of RNA per se in the proteome, as much as the independent role of proteins, has been too long disregarded. When Prasiner observed that some proteins by interaction can change each other's structure, that's the origin of the mad cow disease. They said it's impossible. For 10 years he had troubles. I think he couldn't get tenure even, because it was crazy that something is not guided by the DNA. The same happened for the activities of the RNA. I mean, retroviruses, namely viruses with RNA, were already known, okay, and then they had given some importance to this because they were affecting DNA. But the activity per se of the RNA was disregarded. So your assumption was disregarded by those wrong principles that everything is encoded in DNA. So the reaction, for example, to Kathleen Carrico's proposals to look at RNA functioning in proteomes and perhaps even produce vaccines with it, which dates from 95, was considered crazy. She couldn't get tenure at UPenn. She had major problems because those are things like an RNA world were considered heterodox and nonsense. So yes, I think we should have given more importance. Or in France, Bruno Canard is another guy who wanted 20 years ago to study RNA, what was doing independent of interaction with DNA, doing in the proteome, in particular RNA viruses. He couldn't get financial support for years. So those areas have all been disregarded because of this genocentric central dogma hegemony, which is still very strong. So in a sense, we have been very late in understanding an RNA world, which is surely very important. I would not be able to say if this is the real origin of life or not, but it's surely very important. As any component, there must be a relativity of causes in biology. Everything matters. That's the point, the level of interactions. Some, of course, are particularly important, and RNA is surely one of them, but all levels of organizations interact in producing phenotypes. And RNA is surely very important. We were very late, even though they say, now we've been very fast in producing RNA vaccines. Yes, for 20 years, we have been discouraging people who was working on that. Okay.
Great. Thank you. I think we're going to, Katerina has a question, and then we have another question, and then we have a question in the chat afterwards.
Hi, thanks, Professor Longo. This was fascinating. I have one question. You kept referring to the notion of mechanicity. You used it as a concept that was supposed to, epistemologically underpin or explain what you were elaborating here. how do you understand mechanicity? I'm asking this question because very often if in the humanities, when we discuss technology, when we discuss AI, etc., the very mention of the notion of mechanicity is considered as if, you know, kind of vulgarizing the discussion. And it's very often opposed to the notion of organicity, for example. So do you agree that these two things are opposed in some way? And how do you understand how or how do you use the notion or the concept of mechanicity in your analysis when you talk about, you know, things having to be explained as a matter of mechanicity, for example, or mechanism?
Yeah, that's a crucial question, and it's very delicate issue. On one sense, sometimes some abuses must be allowed. Namely, sometimes I understand my colleagues saying, I want to understand the mechanism, and they really are saying, I want to understand the cause of this, of that, and this is legitimate. Unfortunately, the word is not the right one, in my opinion. So I would like to characterize the word as my specific proposal about the history of formation of the object, namely, an analysis and an object is considered to be mechanistic if it may be seen as constructed by composing elementary and simple components, as all you do in all machines. You take elementary and simple components, you put them together, and you make something very complex. In organisms, typically in embryogenesis, the formation of the organism goes by differentiation. It means you have one cell, the zygote, which is an organism. It differentiates. At each step, it is an organism still, and then by differentiation, first in tissues, then in organs, it gets to be an organism like a child. In a sense, you do not make a child by screwing in the fingers one by one, by putting an eye in a hole. You don't do a child in this way, I can tell you. And that's the major difference, is in the history and the analysis of the formation of the technical or the organismal object. I think it's only a matter of the historical construction. That's what makes for me the difference between a machine and an organism. So the unity of the organism is maintained during the entire construction, and the differentiation is cells differentiating in tissues, differentiating in organs, that at certain critical transitions get functional. The beautiful analysis by Noble in the book I mentioned of the formation of the heart, of the vascular system. At a certain point in the embryo, you have at the same time the formation of tissues that will form the heart, that get to a certain structure and thickness. At the same time, you have a molecular expression that is guided by the microtubules that allows ion to go to a certain length. When the length of the microtubules in the cells is enough, so the ion channels work, and the thickness of the membrane is sufficient, you have a critical transition, and whoop, you have the heart which is formed. So we have simultaneous, there is no composition. During the embryo, the differentiation produces a series of structure that at a certain level of interactions, whoop, it works. And doesn't always work. You know, apparently about 60% in women, 60% of zygote don't get to a birth. So something goes wrong because it's a very complex system. So I understand the difference between a mechanistic description and an organismal one by the history of the object, the history of the formation of the object. Now, yeah, related to that, there is a question here. To what extent have theories of autopoiesis affected the mechanized theories of molecular biology, epigenetics? Well, they've made a major contribution. Unfortunately, in Varela's tradition, more than Varela himself, whom I knew was a fantastic thinker, in the Varelian tradition, they don't give relevance to evolution and history. I mean, autopoiesis is ahistorical, which is very bad. nothing in life can be understood without a reference to time, whether the time of evolution or the ontogenetic time. And I said before, the discrimination between an organism and a machine is the history of its formation. Then in time, of course, in time.
Great. Does anybody have any other questions? If not, I have a question here. I guess it has to do with some of the recent advancements in sort of biotechnology and the kind of scientific imaginary or scientific imagination around, say, for example, CRISPR-Cas9. Because you mentioned very briefly in one of your slides gene editing technologies, and I'm thinking about the way in which there's this kind of the sort of history of scientific practices and the kind of disavowal of
your voice. I've lost most of your connection. I've lost 3 minutes.
Oh no. Okay, so I'm going to start again. Can you hear me now, Giuseppe?
Yes, yes.
Okay. Basically, I'm interested in the way in which sort of recent developments in biotechnology and the kind of scientific imagination around, say, for example, CRISPR-Cas9 and gene editing technologies, because you did mention gene editing as a sort of contemporary analog of some of these mechanistic theories of nature and life and so forth. And CRISPR, what's interesting about CRISPR is the way how it's sort of a very subtle development out of the sort of the problems that face synthetic biology. I mean, if you want to think about synthetic biology as sort of the building blocks, right? CRISPR-Cas9 is this way in which it's subtle, it's context specific, it's targeting, and so forth, and it kind of disavows a lot of the problems of mechanization and allows for a new kind of scientific imagination around what it is biotechnologies can do in the editing of genomes and so forth. And I'm just wondering about this way in which we keep on recasting biotechnologies is able to do, is able to respond to context specific problems, and how it is this, sort of, this kind of well, maybe I'll leave it there. I don't have any further questions. I guess I'm really interested in the way in which these biotechnologies keep on reemerging and responding to these problems, knowing all full well that they have to be able to target, and that the problems in synthetic biology is being able to engineer the whole genome or build an organism from scratch is riddled with problems. So, anyways, I'm wondering if you could just say a little bit about that.
Yeah, again, I mean, per se, first, the early discovery of how some bacteria react to viruses by cutting, fragmenting, modifying their DNA is really fantastic. I mean, there's no doubt there's been a major scientific insight to discover this. Then comes in the wrong theoretical frame. First of all, grounded on the exactness. It's amazing. It is so ideological that they sell us something exact, which is cherry picking. Namely, they do on large numbers, then they choose the cell where it worked. Indeed, they do affect fragments of DNA. And most of the time, not always, they do affect the part that they wanted to affect and modify it by cutting and inserting. But there are always off-targets effect, always. In some expected site, and they discover it, and there are massive evidence about that, but there are much more off-target effects in areas that you do not expect to see those effects. They're very hard to discover. As I said, I've been writing in 9 pages, it's almost finalized, review of Doudna's book, and so I give there lots of references to that. But more than the evidence against, what is absolutely clear is the fake theoretical frame. One cannot base a scientific proposal on terms which are crucial and not defined. She talks of information and it is not clear what the hell of information is it. Is a Turing Shannon on discrete data types? Is geometry of information? There are lots of forms of information. Is the programming? Tell me where is the hardware? Tell me what is the compiler, the operating system. You cannot use this vague framework, bring in a structure of causality, which is Laplacian, as Turing and Schrödinger say, through the Schrödinger says, if we will be able to fully decode the structure of chromosome, and if I'm right that this is the coding of the phenotypes, then we will be in the position of a Laplace demon. We will know perfectly well everything. Then let's go to something different. The second part of the book, he analyzes a very nice point of view about entropy and negentropy in life, which is a totally different perspective. Turing says the same. My machine is Laplacian, namely determination implies predictability. You handle here deterministically and you fully predict the output of a program. If your program doesn't do what it should do, you will get very upset. Programs do work. And if they don't work, what you do, you fire your software designer. I mean, it's inconceivable that the program doesn't do. Even in networks, our colleagues are so good that in spite of the noise on networks, oscillations and electromagnetic fields and atmospheric conditions, there's lots of noise. In spite of that, internet works. If you push a button, it does always the same thing, which is Laplacian. You want to predict what does your command on extremely complex networks, because noise can be eliminated in discrete state machines. It's hard because it's a mess, but it's possible and it works. Now, this vision of life where there is not noise, but there is randomness, and this randomness is a component of the production of diversity. And diversity is an essential component of stability of life. They have a totally wrong paradigm, you see? In physics, one can make a fine distinction between randomness, which is unpredictability internal to the theory, and noise. If you use information theories, you have only noise. As Monod says, evolution is due to noise. Amazing. I mean, you know, you miss what accounts in life, namely the permanent production of diversity, which is the fundamental structure of the stability of an ecosystem, of an organism. Our liver has 50% of aneuploidy in cells, namely 50% of cells of the liver have a wrong number of chromosomes, have lots of mutations. What happens? They produce enzymes in a messy way, which helps, is fundamental to resist the many different shocks, I mean, we have from ingesting the diversity of food. The diversity, the messy diversity of the liver is absolutely essential for the liver to function correctly, not to mention the immune system, which of course works because it produces randomness all the time. So they really have totally wrong paradigm. And that's fundamental, of course, in understanding the role of diversity in the ecosystem. You may also claim that, oh, yes, we understand diversity, but the theoretical frame is the exact opposite, and that makes major damage in our relation to the ecosystem.
So I just have a really quick follow-up because I wanted to, and then I know Vera has a question afterwards. I'm just thinking about, you know, the sort of domains where we could see like the real consequences of something like this. And that would be, I know this is an area that you work quite closely on, Giuseppe, is it's cancer research, right? I mean, I'm thinking about this sort of, especially in the United States, the kind of discourse, the ideological discourse on the war on cancer, which is essentially that we need to, as soon as we identify it, we need to get rid of it. We need to get it out of our system, cut it out, and do whatever we can. But actually, with a lot of those therapies, of course, what you're doing is killing the host. You're killing the organism itself. So I'm thinking about, you know, this sort of the kind of conversations that are happening right now around the sort of evolution in cancer and the sort of co-evolution of cancer. And it's not simply, I mean, you know, cancer's been around for a very long time, and how it is that we co-evolve with this. And this kind of discourse is fundamentally at odds with a sort of mechanistic understanding and a molecular understanding of cancer and how we live with these sorts of mutations?
Yeah, well, that's another very long discussion. Indeed, most of my activity in biology has been in collaboration with biologists focusing on cancer, and in particular on the causes of cancer, namely because the old genocentric perspective, with pursuing the myth of the magic genetic ballot that would cure cancer at the genetic level, disregard the causes of cancer, which has been doubling in incidence in the last 40 years. We know very well the endocrine disruptor, the increase of cancers related to organs mostly and largely regulated by the endocrine system that is disrupted by a variety, we inserted in the ecosystem 80,000 new molecules in the 20th century, 80,000 new molecules. Well, saying, no, they're very different from the organic molecules, so they will not affect the key lock paradigm. They are not stereospecific. They do affect improbabilities, the molecular cascade, the hormonal cascade. They do affect. Bisphenol A is not identical to the estrogens it is close to, it's very different, it's different. It doesn't affect the receptors in the same high probabilities, but with low probabilities. With time, it gives cancer though, because it changes the probabilities. So it's that's another area where situation is really frightening because of the blindness. They keep saying, oh, next year we'll have. In 2000, Van Eijsbach, head of the National Cancer Institute, NCI, writes a paper promising the disappearance of cancer by 2015. Not a cure, that in 1 or 2 years, we were going to have diagnosis and prognosis by looking at DNA, 1 or 2 years from 2001. We all know, because we all have this terrible experience, directly or indirectly, that only the histologist looking at the tissue at a light microscope can say whether it is a benign, a malignant cancer, whether it is a primary or a metastasis. 20 years after. No genetic analysis can tell you about the nature of the cancer. It's amazing. And they've been putting all the money of the National Cancer Institute, 3 billion per year, all the money in that search for the magic genetic ballot. What can we say? But the point is that these guys are still in power. There's no doubt. They're still there. They still handle the financial resources. Yes. Oh, I see Vera. Hello, Vera. I'm pleased to see you. How are you?
Giuseppe. Likewise. I greatly enjoyed your talk, as every time I listen to you. I have one thing that I was, it's maybe a bit of an indirect question, but I have been quite puzzled lately when I learned about this one book by René Descartes, which he called The World, and where he actually writes about an architectonics of the world, which was to me extremely surprising and very hard to reconcile with everything I had learned about Descartes. He says the world is a plenum, but it has cracks from the beginning, and the matter always immediately fills up the cracks. So it's a dynamic model, so that the void never becomes a real dimension. And for me, this is a very virtual kind of projection space for the world, where it feels like mechanism and contingency do not contradict, but need one another at the core of his thinking. But I am not very well familiar with it at all. I'm just starting to research this, but I would be super interested in hearing what you think about that.
Oh, well, the founding fathers are always immense, much better than the followers. So, of course, Descartes is immense, and he explores the other ways as well. The point is the resulting dominating philosophy, which has been extracted, is what has had a major historical impact. But no doubt, yes, him, not to mention Spinoza is another. So yeah, or Hilbert in Foundational Mathematics. Hilbert is great. The Hilbertian that they've been dominating, have been taking up only that fragment of philosophy and turning it into a very limited perspective, the formalists in mathematics. Yes, I know, like Galileo. Of course, all what he says is about life is allometry, which is very nice, but it's purely mechanical. However, he has beautiful pages about his surprise of the diversity of life. Namely, he's very surprised by the fact that from America, there were coming new animals, new plants, and then he says, how comes if God created us at the same time? All that, how is it that there were new ones there, diverse animals? So these guys were fantastic. Yeah, sure. I don't know the pages you mentioned, but I also read beautiful remarks by the founding fathers. We have to go back to them also. Nonetheless, I mean, there was a change. If you go to pages by Leonardo and Cardano, you would see they had a much higher appreciation of what you were mentioning, like turbulence, contingency, dynamicity. For Leonardo, he wants to represent movement. What he cares of is natural movement. That's really what he wants to, and that's why in his paintings he has never lines. He has only degrees of variation of color. He represents everything by degrees of variation of colors, degrees of variation of shadows. That's the very delicate moving of the light. That's what he wants to give us. He needed years to do one painting because we want to accompany without ever making a line. As a very strong fight with me, Michelangelo, because Michelangelo has strong lines like that, and he doesn't like it. It's a very different perspective. That's the movements of light that you want to accompany. Yeah.
If I may follow up on this, can I?
Yes, for me.
Very quickly, because that was the other point. I was hesitating. Of course, I see what you mean by this fascination and this enormous affective sensitivity, that Leonardo with the vortexes and with this kind of it's a kind of an intuitive precision too, So there is a precision, I would say, to it as well. But then I was not easy with the way you put Alberti, as if he was not so interested, because I think what is something that links the Alberti stance with the Cartesian stance, and is different from Michelangelo or from Leonardo, is an architectonic stance, which wants to be synthesizing, so not mimicking, not representing, not understanding, and then approximating, but artificially synthesizing as an artifact. And Alberti has this way of thinking of what a house is, which I find it comes so close to your emphasis on the line, which cannot be there. So on the border, on the introduction of the border, because Alberti says a house is neither painting nor is it sculpture. It is entirely made out of surfaces. So surfaces which come to appear because they're as if painted, but they don't form a painting. They form a sculptural body, but they are not a sculpture because there is no body that it would mimic. And I don't remember now the precise formulation, but it was so delicate and it was super nice, I thought. And it had to do with a similar interest in synthesis, which doesn't aim at controlling and overtaking. It aims at perhaps a little bit outsmarting, on a modest scale. It doesn't need to mean conquest. And I was very fond of that aspect.
Oh, but sure. Now, I only quoted Leonardo disagreement on this specific issue, namely for Alberti, who understand the relevance of having lines with no thickness, like Piero della Francesca, also Piero Francesca, he understands that it's a core idea in Euclid. And he says, how can I, as a painter, draw a line with no thickness? I cannot yet. I will do the tiniest line I can, but I understand that mathematically it must have no thickness. And so Alberti says the line is made out of points. And then all what you said is surely correct. I don't know anything else. I only know that Leonardo disagrees in order to go further by saying the line is a movement of a point, which I find beautiful. And the plane is the translation, is the movement of a line, which is again a more dynamic perspective in the sense that seeing the plane is made out of lines. So this I only quoted Leonardo's, I don't know if polemic. No, the lines is not polemic, simply saying, I don't agree with Alberti, I think that. Yeah. So that's the only disagreement.
But I think it's not a disagreement at all, because with Alberti, then it becomes color. So the point in movement, which gives the line, is not in him resolved in lines anymore, but becomes surfaces, so painted surfaces, appearances of colors. But then again, but I very much liked this very abstract and so precise, precise pointing us to lines that are so consistent and so relevant with regard to AI questions and big data and cipher keys as well. Also, there would be another question, but for another time maybe.
Okay, thank you.
All right, we have Devor, you have one. You have a question. I think we'll have to make this the last question.
Okay, thank you very much. I'll make it short. Professor Longo, you have indicated that we are facing a scientific revolution, right? Many times. So you said to understand genes correctly will be similar, a shift like from the Ptolemaic worldview to to the geocentric, heliocentric worldview. And I would like to ask you if you could elaborate a little bit on this in regard to materiality. So the classical, let's say 20th century classical perspective on genes was, as you described, like a program, right? We have like an alphabet, like we have the key lock metaphor and the hand glove metaphor, so to speak. So you have a program of genes that are translated into enzymes and proteins and so on, and then they make a body. Yeah? And all this is happening, so to speak, on the basis of chemistry. Now with the new perspective, with this processual perspective that you're indicating, where the context plays a role, where we don't have this mechanistical causal programs, is it still based, so to speak, on chemistry? So is the materiality of how should I say, of the causalities or the ontology of these causalities, is it still chemistry? Is it still physics, or is there some new force, some new causality appearing, which cannot be mapped by the classical positivistic sciences like physics, chemistry, So you see how deep you think is this revolution going to go? Are we just shifting our perspective on biology by taking more chemistry into account, to see new mechanisms in chemistry, or do you think there is some sort of new or different force appearing?
Well, it is a broadening of the structure of determination, in the sense we have to take sort of noble as the right wording of biological relativity. All level of organization have a causal role in a biological process. So the only way to understand a molecular network like those that exist in a cell and that are physically impossible. Namely, if you take these huge molecules interacting these huge networks, and you consider the possibility of having them poorly by physical composition, the probabilities are almost zero. The molecular networks are made possible only under the constraint of an existing cell. That's why there is no life emerging every day, because only life can come from life. You need the constraint of a cell, the membrane, the internal structure of the cell in order to make possible those molecular networks that produce the constraints, because those molecular networks do produce the cellular structure, the membranes, and so on and so forth. So all those levels concur at the same time to make life possible. And of course, there are physical-chemical processes, and you know physical chemistry, which physics is a problem because there are both. I've been working with the genetician of plants on the role of quantum phenomena. There is a broad literature. There are plenty of quantum effects that are crucial for the activity of cells. Both quantum randomness and classical randomness mix up in a way that physics cannot handle, because physics cannot handle at once the classical and quantum fields, and nor classical and quantum randomness with entanglement effects, and they live together in cells under the constraint that they themselves produce those processes. That's why it's the immense complication. That's why there is an idea in autopoiesis, which is very, very useful, and it's part of a frame. We have to invent a proper causality that brings simultaneously together all these different levels of interactions with no priority. That's the major challenge. Focusing on the origin of life, we have simply no idea, because it's a process where many levels of causalities are all necessary to allow the process itself. Again, no way you can see in physics these incredibly big molecules in incredibly complicated networks that can only exist in a living cells. That's the amazing structure. The enablement is a crucial. Only the cell enables those chemical structures. Can you study it then with pure chemistry? No, because physics, no. You need to allow a causal thinking that includes the constraints, the membrane, the organismal constraint, the context that gives the pressure and the torsion on the chromatin, the structure of the chromosomes, modifies the expression. So they are causally relevant to the DNA activities. There's nothing to do with information. If you put pressure and torsion on your computer, you do not contribute to the elaboration of information. I mean, well, it is essential in the chemical activities. So we need the strong extension of chemistry and physics. An extension in a logical sense. You need proper properties and principles that add on top and interact with the principles that we already have for chemistry and physics. In logic, there is a beautiful notion, being conservative extension or not. Conservative means that if you add extra principles, however, you do not prove more properties of the simpler level. I think that the proper biological theoretizing is a non-conservative extension, namely that allows to prove chemical physical properties that you cannot prove by purely chemical physics. I think that, again, because the purely chemical physical networks existing in cells could never be derived for purely physical chemical reasons. They have probability 0. They are so unlikely. Almost 0.
So you're saying back to emergence, to the concept of emergence.
Again, if I don't like that concept, in a sense, some people say, for example, the property of water emerged from quantum properties. No, it's the theory that changes. Namely, we need hydrodynamics of incompressible fluids, which cannot be understood in terms of Schrödinger nor Boltzmann. Incompressible fluids deserve its own theory that deals with incompressible properties of water. I don't think that it's right to say that the properties of incompressible fluids emerge. No. We change theory. Then the challenge is to unify, to give a theory that brings together Navier-Stokes equations for incompressible fluids and Schrödinger or Boltzmann equation for atoms and statistics for quanta. You see, that's a different attitude. I'm not against, but I don't use the word emergence. I'm not against, again, because most of the time it's simply that we are describing the phenomena at different levels with different theories. emergence often is synonymous of that. Again, I'm not against, but I think it's scientifically ambiguous.
Okay, thanks.
Great. Well, I want to thank Professor Longo for his amazing talk and contribution to the SMR. So I think we're going to wrap up now. But please do stay tuned for our next presentation, which is by Alexander Wilson, titled Non-Real Patterns in the Automation of Science. And that will be on the 25th, which is just in 2 days. So thank you so much, Professor Longo, for your time and contribution.